Now for the next stage, we are going to do translation. We're going to take this mRNA, which is a copy of a DNA gene, and we're going to make a protein from the information here. So what happens first is we've got to get a ribosome clamped onto this mRNA. But first things first, we're going to bring in a tRNA or a transfer RNA that matches this first one. So my tRNA is going to come in, and it has an anticodon that matches this. So remember, we don't have any T's for RNA, so this is going to be a UAC. Now, this is where I have to go to a codon chart. You have this in the book. And I'm going to look for AUG. Now, I'm not going to look for the UAC because that is an anticodon, and this is a codon chart. So I'm going to look for AUG and see what that spells. So, here it is, AUG. That spells MET. So we have a MET on there. Now, the ribosome is going to clamp over the top of this mRNA, and it's got 3 spaces. And they are the E, P, and A sites. And then we're going to clamp on to the bottom of this the rest of the ribosome. And we're going to go ahead and label this ribosome, and this is the mRNA. So now I've got one tRNA on there, and I need to add another one. I need to add another tRNA that's going to match this CUA. So what goes with CUA is going to be GAU. And pulling out my handy-dandy codon chart, I'm going to look for CUA. So here is CUA, and that's going to be leucine. By the way, do you need to know the names of the amino acids? No. But you do need to know how to use a codon chart. Okay, so the next thing that's going to happen is this ribosome is going to move down 1 space. And in the process, it's going to hook this methionine onto this leucine. So I'm going to pause, and I'm going to fill in my drawing, and we'll go from there. So the ribosome has moved down the mRNA 1 space so that this that used to be here in the A site is now in the P site, and we've got our first tRNA. It's lost its amino acid. It's been moved over to attach to the other amino acid. So now I have 2 peptides that are hooked together. So the A site is now open, and a tRNA from the outside that matches this, so it's going to be UAC again, so it's going to be another methionine. No, you don't have to have these memorized. That's just one that I happen to know. So it's going to come in, and it's going to bind here, and we're going to have MET here. Now, this one is empty. It doesn't have a tRNA, so it's going to go out the E site. It's going to exit out the E. Now, this ribosome is going to move down 1 more, and this leucine is going to attach to this methionine. And the ribosome is going to keep marching down the mRNA, and it's going to keep adding amino acids based on the code that's in the mRNA. So I've run out of room, so I'm going to erase this, and I'm going to have the toward the end of the translation process. So now we are at the end of our mRNA. Our ribosome has marched down, and we've hooked this growing strand of amino acids, which we call a protein, over onto this tRNA, and it's going up out of the ribosome, and it's folding into its proper shape. So this tRNA that coded for ARG is going to go out the E site. Here's our P for peptidyl, or I think of it as the protein part. And here's our A for the addition site. So now we are to the last codon, and it is UAA. And UAA codes for stop. That means there's not a tRNA that's going to match that codon. So what happens instead is that signals, actually we have a protein that comes in here, that comes in and signals the ribosome to open up. These tRNAs detach from the mRNA, and this ribosome can go back to the beginning and clamp down and start over again. And it can make another protein from this mRNA, and this protein is going to detach, and it's going to go off and do its job. So if it's an enzyme, it's going to start doing its job. If it's a structural protein, it's going to be put in the cell where it belongs, and that's translation. So that's what the cell does with the energy, the ATP, that is generated by glycolysis, fermentation, the Krebs cycle, you name it.